In today's lecture, we will talk about respiration. And what you see in the background of this slide are two mitochondria, here and here. These both are from human cells, specifically human lung cells. Plants also have mitochondria, and we'll see in a bit their importance for the process of aerobic respiration. In today's lecture, we'll start by reviewing some information we said previously in the metabolism lecture. First, we'll go over the general formula for respiration and compare it to photosynthesis. Then we'll talk more about the relationship of photosynthesis and respiration. In terms of chemical formulas, they are opposite. However, they are also complementary in that the plant needs to do both in order to survive. Next, we will go over the steps in respiration. No matter what respiration will start with glycolysis. The next steps will depend on whether there is oxygen present or not. If oxygen is present, then the next important organelle will be mitochondria, and this organelle will be where the next steps occur. We'll talk about the origin of mitochondria, which come from endosymbiosis. This is similar to how chloroplasts became part of cells. Within mitochondria, the two next steps of aerobic respiration are the citric acid cycle and the electron transport chain. Let's just specify aerobic respiration has those two steps. In anaerobic respiration, fermentation will happen instead. Aerobic means with air, but really it means with oxygen. Anaerobic means without air. Really what it means is without oxygen. We'll talk just a little bit about fermentation, and then we will talk about the implications of plant respiration for humans at the end. We will start with review. Respiration, in terms of the chemical reactants and products, is the reverse process of photosynthesis. This is because glucose is broken down into carbon dioxide and water. Remember that glucose was a product of photosynthesis, and the two waste products of respiration, carbon dioxide and water, were the two ingredients for photosynthesis. In the process of respiration, energy is released, and the process requires oxygen. Let's talk a little bit about how the respiration of glucose is really an oxidation process. Remember that oxidation is loss of electrons, reduction is gain of electrons. We'll focus here on aerobic respiration. In anaerobic respiration, the same things that we'll talk about are true, but to a lesser degree. Focusing on aerobic respiration, we will see that glucose is going to give up hydrogens, these H's here, and by the end, it is going to become carbon dioxide. So focusing on the loss of these hydrogens, if they are being lost, that means each time one is taken away, one proton and one electron is removed. This is because a hydrogen really is just one proton plus one electron. Remember that oxidation is loss of electrons. So if glucose is losing hydrogens in this process, it's losing electrons, and therefore it is becoming oxidized. Well, electrons never disappear, so they need to go somewhere. Where do they end up? It turns out that in the process of respiration, both the electrons that are pulled off and the hydrogen ions, or the protons, they end up attached to oxygen. This means that the oxygen is gaining electrons, therefore the oxygen is becoming reduced. And in this process, once oxygen gains both protons and electrons, it's really gaining hydrogens, and therefore, once it gains the appropriate number, oxygen will become water. We've seen the equations for respiration and photosynthesis before. The top equation here is for aerobic respiration, the complete breakdown of glucose into carbon dioxide and water. And this process, as we've already said, requires oxygen. So the two reactants are glucose and oxygen. The two products are carbon dioxide and water. As I mentioned in the previous lecture, this equation is not balanced. It's not telling you how many of each, it's just telling you what each of the categories are. Remember, in photosynthesis, just for a reminder, the process is the reverse. So our two products up here for respiration are the two ingredients for photosynthesis, carbon dioxide and water. The two ingredients for respiration, glucose and oxygen, are the two products of photosynthesis. That makes these two processes seem like opposites. And chemically, to a large extent, that's true. The processes are a little bit different, but at least the reactants and products are opposite. However, these two processes nevertheless work side by side in plant cells. And the reason for this is that chloroplasts are good at capturing sun energy, but they are not good at making ATP available for the cell. So instead, what's going to happen is the chloroplast is going to focus on capturing energy from the sun as well as carbon dioxide and using it to make glucose. That glucose then will be made available to first reactions in the cytoplasm, and then later products from those reactions will be made available to the mitochondria. And this is the location where additional reactions will occur and the chemicals will be fully broken down to make carbon dioxide. In the process, a lot of ATP is going to be made. And that ATP made in the mitochondria then is sent out into the cytoplasm of the cell, represented by the blue out here, to be used for a wide variety of cellular functions. So these two processes are not opposing each other. Instead, this process happens, it makes a product that is eventually used by the processes in the mitochondria, and that then powers what happens in the cell. This is review. We've already talked about ATP, but it will be important. So ATP, remember, is a cellular compound used to store energy. And we've said it's only good for short-term energy storage, which is why the plant instead makes glucose when using sunlight and performing photosynthesis. ATP powers many cellular reactions. It occurs in plants, bacteria, animals, and all other groups that I am familiar with, at least. Remember that the low energy form of this is ADP. So when ATP becomes ADP, energy is released, but to make ADP into ATP, a source of energy is required. This is the high energy form. Our last general reminder is that some reactions in both photosynthesis and respiration will require some molecules to carry electrons around in the cell. And I'm referring to these simply as electron carriers. We said that in photosynthesis, the most important carrier is NADP. I pretend this P stands for photosynthesis. Chemically, that's not true, but it makes it easy to remember. The most important carrier in respiration is NAD. There's at least one other, but this is the primary one. We're now going to get into new material. So we are going to break down the process of respiration into 3 steps for aerobic respiration. We'll talk about an alternative end step in anaerobic at the end of this lecture. In aerobic respiration, in either process, whether aerobic or anaerobic, the first step is always glycolysis. Glycolysis will occur outside of the mitochondria in the cytoplasm of the cell. So let's look back briefly. If this is our cell, and this is our mitochondria, then the process of glycolysis will happen somewhere out here. The process starts with glucose, which is represented up here. I'm only showing the carbons as blue balls. There would also be oxygens and hydrogens attached. And remember that glucose is a 6-carbon compound, so there are 6 of those balls represented here. During the process, several changes will happen, and I'm greatly simplifying compared to what the book tells you about this. But by the end of the process of glycolysis, there will be, instead of 1 6-carbon sugar, there will be 2 3-carbon compounds. That means none of the carbons have been lost. There were 6 up here, there are 6 down here. But now it is 2 different compounds, each with 3 carbons, instead of 1 with 6. That's the first major difference. The second is that there is a net gain of ATP. So each time this happens, there's a gain of 2 ATP per glucose made. There is also a reduction of 2 of these electron carriers from the form without the extra electrons and proton to the form with them. That means there's a change from NAD plus to NADH, which has the extra electrons. There will be a gain of 2 of those each time glycolysis happens for 1 glucose. Here are our 3-carbon sugars. We've said there's 2 ATP, 2 NAD plus are reduced to NADH. We are ready to go on to the next step. It's important here that the next step will only happen if oxygen is available. Therefore, we are talking about aerobic respiration. This is where mitochondria will become important. Let's talk a little bit about where they come from. If you recall, chloroplasts came from a process of endosymbiosis, where a free-living bacteria capable of photosynthesis was engulfed by another cell, and the bacteria, instead of dying, became a long-term partner, and it evolved from being a free-living organism to being an organelle of the cell it entered. Exactly the same thing is true for mitochondria. They were historically free-living bacteria that were not part of eukaryotic cells. But at some point, one got engulfed, and when it became engulfed, instead of dying, it continued to reproduce and live within the cell. And as the cell reproduced, the mitochondria would reproduce too. Over a long period of time, there was opportunity for evolution, and the bacteria lost some of the traits of free-living organisms, and it gained better abilities to coordinate with the cell. So it went from being a true bacteria to now just being an organelle that is historically related to bacteria. We will look at the structure of a mitochondria. Really, the singular is mitochondrion. There are 4 parts of this I want you to be familiar with. Around the very outside is the outer membrane that is shown in the orange color out here. Then there's a space and another membrane. This additional membrane is much more wavy with these infoldings, and this is called simply the inner membrane. The space between the inner membrane and the outer membrane, so this whole space, is called the intermembrane space because it's between them. Finally, the name for the space inside the inner membrane, it's called the mitochondrial matrix. And this is where many of the important reactions will occur that we will discuss. The first step of reactions that happen inside the mitochondria are called the citric acid cycle. So let's go back and remember that glycolysis ended with 3-carbon sugars. Those 3-carbon sugars are sent from the cytoplasm out here into the mitochondria. Once they go into the mitochondria, then a series of reactions will occur. these will be, or they will include oxidation reactions. That means electrons, as well as some hydrogens or hydrogen ions, will be pulled off. And additionally, carbons themselves will get removed. And eventually, all three of the carbons will become separated. That means the 3-carbon product is completely broken down until all that's left of it are 3 separate carbon dioxide molecules. So the result of the citric acid cycle is production of carbon dioxide, really 3 of these for each of those 3 carbon compounds that start the process. This is not being made because the plant wants it. This is really just a waste product that will get released into the air. There's production of a little bit of ATP. Remember that a little bit of ATP was made in glycolysis. A little bit more is made here in the citric acid cycle. More importantly, there are going to be 4 electron carriers, and 3 of these 4 will be NAD. The other one we won't worry about by name. But all 4 of these will carry electrons to another stage in this process that we'll talk about in a minute. And we'll see this is really important because almost all of the ATP that's made by the mitochondria in this process is really made using the energy stored on those electrons that are attached to the electron carriers. We'll talk about that next step now. This step is called the electron transport chain, and it will be very similar to how we saw ATP was made by chloroplasts as an intermediate step in the process of photosynthesis. The electron transport chain is going to occur with proteins that are embedded in this inner mitochondrial membrane with all of the infoldings. The infoldings basically give this membrane a lot of surface area, so a lot of these proteins can be attached. This is what the process looks like more up close. And as you can see, it's a little bit involved. The important thing to get here is that in the process of the electron transport chain, electrons are going to get passed among proteins that are sitting in the mitochondrial membrane. As those electrons get passed, hydrogen ions are going to get pumped. So the electrons carry energy, they release some of that energy, and that powers pumping hydrogen ions into the intermembrane space. That's where they'll be stored. In photosynthesis, we talked about this being like a battery. The same thing is true here. We'll talk more about that in just a minute. The first step here is that electrons from electron carriers are passed to these 3 biggest proteins sitting in the inner mitochondrial membrane. So you can see this here. Most of the electron carriers will drop off their electrons at the first member of this set of 3 in the chain. One of them will drop off their electrons a little bit further on. We won't worry much about that distinction. Next, the electrons are going to get passed from one protein in the complex to the next to the next. So you can see this path here. Electrons go from here, then they are carried by a small carrier. They end up at the second major part of the chain. They're carried by a second small carrier, and they end up at the third part of the chain. Each time they are passed, hydrogen ions are pumped. Now, at the very end, the electrons need to go somewhere. So first let's say, the hydrogen ions are pumped into the intermembrane space. This creates a difference in hydrogen ion concentration across the membrane. A high concentration up here, a low concentration down here. Where do those electrons end up? They can't just end up getting given off into space or being let loose into the mitochondrial matrix. Instead, they have to be put onto something. So at the end of this chain, the electrons are attached to oxygen. When the electrons attach to oxygen and some of the remaining protons stick on, then the result of that is this oxygen becomes a molecule of water, or H2O. We now have a situation where this difference in hydrogen concentration has stored energy. This is basically a battery, and we'll talk about how this can be used to release the energy and power production of ATP. Just like we saw in photosynthesis, the hydrogen ion gradient will power an enzyme called ATP synthase, pictured here as this aqua shape. And these hydrogen ions in the space between the two membranes, so they've all been pumped into this narrow space that I'm highlighting between the outer membrane and the inner membrane. They want to escape from that space. They come through ATP synthase that releases energy. ATP synthase uses that energy to take low energy ADP and make higher energy ATP. We now have our source of ATP for the cell. It turns out that each time one of the electron carriers drops off electrons, then there are multiple opportunities for this electron transport chain to pump hydrogen ions. And the more hydrogen ions that get pumped, the more ATP will be able to be made. We'll look at the two parts of this process together. The first part was the electron transport chain itself. Remember, in that, electron carriers drop off electrons. Those electrons get passed around. Hydrogen ions get pumped. Then those electrons are given to water, which would happen around here. They're given to oxygen and they make water, is more accurate. These hydrogen ions then come back through ATP synthase, and as a result, you can see in the background of the video that ADP is made into ATP. It's important to note here that the process of the electron transport chain is really where almost all ATP in aerobic respiration is made. This process involving accumulating hydrogen ions and letting them escape through ATP synthase produces about 5 times more ATP than glycolysis and the citric acid cycle combined. This means that if our cells lack oxygen, we are going to lose the ability to make almost all of our ATP. We'll be keeping only around maybe about 20% of our ability. And having 20% of our ATP is not a good situation. This is why it's really important that we continue breathing and taking in new oxygen. The same thing is going to be true of plants. They need a continuous supply of oxygen so they can maintain the electron transport chain, so they can make a lot of ATP. Nevertheless, there are situations where simply there is no oxygen present. Maybe the plant is in soil that's too wet, for example. Under those situations, then it is not possible to do the electron transport chain. Instead, an alternative process is needed. That alternative process is fermentation. Fermentation then occurs under anaerobic conditions, oxygen absent. This process still starts with glycolysis. So glycolysis will be universal for both plants and animals. However, after glycolysis, if fermentation occurs, then neither the citric acid cycle nor the electrons transport chain will occur. Instead, an alternative chemical pathway occurs, and the result of this pathway, in plants at least, will be the production of alcohol. This is the same kind of alcohol that would be consumed by humans. And there's also production of a little bit of carbon dioxide, not as much as in the citric acid cycle. Now, the process of fermentation in plants is the same for our purposes as the process that occurs in a fungus. We use the process of fermentation in yeast specifically for a couple of different uses as people. We use it to produce alcohol for consumption. We also use it in the process of making bread. It is the source of the carbon dioxide that is used to make bubbles, and those bubbles are what make bread rise. There are a couple of problems with the process of fermentation. The first major problem is that it does not result in any additional ATP beyond what was made in glycolysis. So there's no additional ATP beyond glycolysis. This means that the plant is going to lack a good source of ATP and it will be stressed and may die. The second problem with this is that the alcohol that's made is toxic. Just as alcohol is toxic to humans, especially in large amounts, it will be toxic to plants and other organisms. Therefore, this process really is not ideal. It is sort of a better than nothing, but plants prefer to avoid it if possible. Let's talk about what happens if we overwater plants, and we'll relate this to respiration. Remember that if water is added to soil, especially inside pots, or other soil that doesn't drain very well, then the water is going to displace the air pockets in the soil. Of course, the air pockets are where a large amount of oxygen is going to be. This means that overwatered soil is going to lack oxygen. This is especially true if you are growing plants in a pot that don't have any drainage holes. The plants that we are growing coleus in, for example, do have about 4 holes arranged around the bottom. That is to allow extra water to escape and to avoid this problem. If you have any plants at home that lack holes, you need to be extra careful not to overwater them. If the plant is overwatered, then because there is a lack of air, there's a lack of oxygen. This means that the plants will have to perform fermentation in their roots instead of aerobic respiration. But if they perform fermentation, then they are going to lack ATP, and this can lead to root death. But root death means that the roots are not going to be able to acquire as much nutrients. If that's true, you'll eventually see it in the leaves. So what we can see here is a plant that has yellowing leaf tips. Yellowing leaf tips, especially at the end of branches, is often a sign of a nutrient deficiency, and this can be caused by the roots themselves being in bad shape. How can we avoid overwatering? Well, first, don't water too much. Second, watch for soil that feels soggy to the touch. Also, if you see algal growth on the surface, that's a sign you might be keeping it too moist. If you do detect overwatering, move the plant to the shade. That's counterintuitive because the sun would help water evaporate faster. The problem is that if the roots are not working well, then the plant will be stressed, and a stressed plant will not be good at acquiring water from the soil, even if it's abundant. Therefore, the plant in the sun will be stressed and not able to acquire enough water to deal with it. So put it in the shade. If possible, remove the plant from the pot. This will allow water to evaporate from all sides of the soil, and it can dry out faster. It also gives more access for oxygen to diffuse inward. You may want to break apart the root ball of the plant. If you find rotting roots, then they would need to be removed. Stop watering until the roots are of a reasonable dampness or the soil is reasonably wet, and then the plant can be returned to the pot. If it's a pot that did not have drainage holes, then replant it into one that does, or create some drainage holes yourself. Last, we will talk about the effect of respiration on the quality of produce, or fruits and vegetables. Remember that respiration is a process that breaks down stored energy reserves over time. That means that the longer fruits and vegetables are stored, the more of their energy reserves are lost. This isn't great for us because when we eat plants, we typically want the energy that's stored there, or at least that's part of why we eat them. So respiration results in reduced food quality. We can slow this process by refrigerating. That creates a colder temperature, and with a colder temperature, the process of respiration happens more slowly. Respiration will directly affect carbohydrates stored in the plant, so sugars or starch. However, other nutrients like proteins can also be impacted because there are ways that the plant can take something like a protein and do some chemical reactions that will make some of its energy available for the process of respiration that we've talked about. So not only will the plant lose carbohydrates, it can lose protein and vitamins, etc. One argument for this is to buy produce that has not been sitting around for very long. Fresher produce is better. Some people suggest that buying produce locally is a good way of achieving this. Secondly, make sure you keep produce cold and make sure that you eat it soon after purchasing or growing it when that's possible. We will end with some final thoughts about respiration. Remember that plants do respire. It's a common misconception that because plants perform photosynthesis, they do not perform respiration. But we've seen that even a cell actively undergoing photosynthesis needs to perform respiration so that ATP can be made available to that cell. Along these lines, remember that plants have both chloroplasts and mitochondria. Animals, of course, do not have chloroplasts. They do have mitochondria. Plants have both. Remember that part of respiration happens in the cytoplasm, but 2 of the 3 important steps of aerobic respiration happen, in fact, inside the mitochondria. And remember that both for plants and animals, which include us, aerobic respiration produces many more times ATP than does anaerobic respiration. So make sure that you keep breathing, make sure when you're exercising that you are getting plenty of oxygen. It will be important for keeping your cells functioning optimally.